Hello, my name is Frederik Hansen and I am a postdoc at the University of Oslo's Department of Pharmacy in Norway. And here I do research into the sample preparation technique called electromembrane extraction, which I will give an introduction to in this presentation. So, electromembrane extraction, which also is abbreviated EME, is what is called a micro-extraction technique, meaning that we generally use quite small volumes of both sample and organic solvent. This technique is derived from the classical liquid-liquid extraction, and essentially this is a process of electrophoresis across an oil membrane. The basic principle is fairly simple, as illustrated in this figure. What can be seen here is that we take a conductive container, in this case a conductive vial, and we load our dirty sample. This could, for example, be a blood-based sample with the analytes of interest into this container. Then we use something called a supported liquid membrane, or SLM, to separate our sample from a clean acceptor solution. This supported liquid membrane is simply a thin piece of polymeric membrane into which we have immobilized a drop of oil or organic solvent, if you will. And then this solvent creates a barrier between the two solutions. And if we then imagine that we have a cation analyte, meaning that it has a positive charge, we can stimulate the migration of this to go into the SLM and onto the acceptor solution by means of an electric field by coupling a voltage to these conductive vials. Now, this basic principle was invented all the way back in 2006 here at University of Oslo. And to date, more than 450 scientific papers have been published. One major limitation of all of these previous works, however, has been that there has not been any commercial equipment to perform this kind of extraction in. However, now commercial equipment is emerging from Extraction Technologies Norway. And this equipment is illustrated here, called ETN12, which we will go into details with momentarily. A frequently asked question for people who are new to this technique is, what can it be used for? And in principle, I will say that any molecule that can have a charge is applicable to electromembrane extraction. And considering that most molecules that exist in the world will be acidic or basic to some extent, then electromembrane extraction is applicable to a large number of substances. If we go a little bit into what it currently is mostly used for, then right now, mostly biological sample matrices such as blood-based or urine or even tissue samples is what is mostly focused on. But all other kinds of applications, such as more environmental applications like wastewater analysis or even extraction from reaction mixtures, is also possible. Considering the biological samples, then of course pharmaceutical drugs is of interest. Endogenous metabolites and to some extent also peptides are possible to extract. And for other kinds of applications, preparative extractions, where you maybe have a goal of removing something from your sample, are also an option. And alternatively, heavy metals can also be extracted with electromembrane extraction. Just to explain in a little more details how we originally came from liquid-liquid extraction to electromembrane extraction, I want to show a few slides. In the classical liquid-liquid extraction, we perform the extraction by having neutral analytes that are present typically in an aqueous sample. Then we put an amount of organic solvent into this, and then we extract the neutral substance into the organic solvent by means of shaking and partitioning. The advantage of this is that it is relatively fast, can be done in 5 to 10 minutes. Disadvantages are, of course, that you use a fairly large amount of solvent, at least 1 to 2 milliliters of solvent per sample. And this technique is not particularly suited for polar or hydrophilic analytes, since that they cannot partition usually into the organic solvent. And also, if you do chromatography afterwards, reverse phase chromatography, you almost always have to do an evaporation and reconstitution into an aqueous solution for it to be compatible with reverse phase chromatography. Alternatively, of course, you can dilute your sample, which also is unattractive. To improve on some of these disadvantages, then some time ago, the concept of liquid phase microextraction was introduced. And in this principle, you take advantage of this supported liquid membrane concept. Now our sample is adjusted to have have neutral analytes, which can then partition passively into this thin barrier of organic solvent. And once they pass through and reach the acceptor solution, then the pH of the acceptor solution is made so that the analyte will gain a charge, which causes the equilibrium to shift towards the acceptor solution, meaning that analytes become trapped here. Just to mention that this supported liquid membrane itself is very thin. It's only about 100 micrometers thick, meaning that the transfer through the liquid membrane is actually fairly fast. The disadvantage of this technique is that we sacrifice some speed, meaning that extractions can take as long as 60 minutes. However, we can reduce the amount of organic solvent we use considerably. So typically we use between 3 and 10 microliters of solvents. We still cannot extract hydrophilic analytes very well, but we do have the direct compatibility with reverse phase chromatography and mass spectrometry, in the sense that our acceptor solution is aqueous, so no evaporation and reconstitution. In order for this to work a little faster than previously, the extraction was tested if it could be assisted by applying a voltage, which then makes the concept into electromembrane extraction, as I explained before. By including the voltage, then we now can do the extraction considerably faster, because the transfer is now actively driven by an electric field. We still have the advantage of using very low volumes of organic solvents, and also the electric field enables us to extract more polar and hydrophilic substances than is possible with the former technique. And we also still have the advantage of the acceptor solution being aqueous and therefore directly MS compatible. If we take a look at some of the other advantages of electromembrane extraction compared to the traditional and alternative sample preparation techniques, then in my opinion, one of the main advantages of this technique is that it is a one-step sample preparation procedure, meaning that we can go from something as complex as whole blood to a clean acceptor solution that is purified of matrix components in just one single step, without having to spend time on transferring and evaporating and things like that. Additionally, as mentioned, it is compatible with very complex samples ranging from whole blood to plasma or serum, or urine, or cerebrospinal fluid, or even tissue has been extracted previously. The technique offers selectivity that is based on both the analyte charge and hydrophobicity, in the sense that only substances that have the right charge will migrate in the right direction in this electric field. For example, here you can see that the cation will migrate towards the negative electrode, whereas the anion will stay with the positive electrode. If I'm interested in the anion, then I just flip the voltage and extract it in the other direction. And this liquid membrane solvent also determines what kind of analyte characteristics are extracted, and this generally is linked to hydrophobicity, which you also will see. But this means that the extracts we get here in the acceptor solution are quite clean and pure, which of course is an advantage for subsequent analyses. This involves the complete discrimination of proteins and phospholipids, which both are quite troublesome when you run reverse phase chromatography and mass spectrometry by clogging the column or causing ion suppression. But these we can eliminate. Other techniques, other advantages include that this technique in principle is non-destructive, since you can reuse your sample after extraction, and also because we use a very low volume of organic solvent, and also it's quite safe solvent. This represents really green chemistry, which is a really upcoming area of focus, both among actors but also among regulators. If we take a look at what this new commercial equipment looks like, then as you saw from the principle sketches before, then this is based on conductive containers that here are shaped in the form of classical HPLC vials. These vials are made in a conductive polymer, that's why they are black. And you can see here that we have two different sizes that are available. One is 200 microliters and one is 600 microliters. These are screwed into a leak-tight interface or union, and in this interface we will have a polypropylene membrane, as you can see here, and this will then hold our solvents. The working volume for these vial sizes is generally, for the larger one, between 200 and 500, and for the smaller one it's 30 to 150 microliters that you can load into. And this means that you have the opportunity to do pre-concentration. For example, if you use 500 microliters of sample going into 50 microliters of acceptor, you can gain up to 10-fold pre-concentration. To prepare for an extraction, you simply take your sample and your acceptor solution and load them into respective vials. Then you place this polypropylene membrane into this leak-tight interface and you fasten it to the acceptor vial, as can be seen here. Then you pipette approximately 10 microliters of the organic solvent onto the surface of this membrane, which then will go into the pores of the membrane. And as you can see from this illustration, it's quite clear when you have done this correctly, because the membrane will turn from white to transparent. Then you simply fasten the sample vial into the interface and place this now EME unit into a shaker, where you apply a lid and then you apply shaking and voltage and wait for a certain amount of time. The current equipment is capable of holding up to 12 simultaneous EME units. As you can see here, these are placed in a holder, and then the lid will have one electrode touching each vial, so we can close the electric circuits. Extraction is initiated by applying the voltage and also by applying the agitation. So as you can see here, this part of the instrument will shake with some 100 RPMs, and the extraction typically is performed for 15 to 30 minutes, although it can be from 5 to 60 minutes depending a little bit on the volumes you use and also the kind of substances that are extracted. After extraction, you simply disassemble the equipment and you can place a cap on the acceptor vial, as so, and this can then go directly into the HPLC autosampler where the injection can be performed. So you can go very quickly from, again, a very complex sample into something that is going into your autosampler. Right, so now after we have had a bit of an introduction, let's take a look at the methods and the method development that is performed in electromembrane extraction. Generally, the key parameters to optimize are of course sample and acceptor pH, since these determine if your analyte has charge or does not have charge. In addition, the agitation rate is important, since this determines the movement of solutions inside of the vials while the extraction progresses. The vial size and the volumes is chosen according to the needs. For example, do you need the extraction to give pre-concentration, or do you just want it to be fast? Extraction time and voltage also are parameters, and then one of the main parameters is the liquid membrane solvent, as mentioned before, because this essentially determines what is extracted and what is not extracted. Several of these parameters can interact with each other, and therefore we generally recommend to use design of experiments methodology for optimization. Although, of course, you can always do one factor at a time, which also works well in most cases. If we take a deeper look into the kinds of organic solvent that we use, then in general, good EME solvents are first of all water immiscible, meaning that they stay in place inside of this liquid membrane. And also they are non-volatile and non-toxic. And this really is a key in terms of making this green, since we use a very small amount, and also they are very safe for the operator. Additionally, they are slightly conductive, meaning that we can establish an electric field across this thin liquid membrane. And as mentioned, they offer selectivity based on the analyte charge and hydrophobicity. And because that these criteria need to be fulfilled, then the solvents we use are slightly different from classical liquid extraction solvents, such as chloroform or hexane, which also is sometimes used. And one example of the solvents we use is 2-nitrophenyl octyl ether, which is abbreviated NPOE. This solvent is capable of extracting bases, or meaning positively charged ions or cations, with a log P of about 2.5 to 6. This range is illustrated in the figure you see here, where you have the extraction recovery, meaning of the substance that is present in the sample originally, how much is transferred into the acceptor solution. So 100% means everything is transferred. This is plotted against the log P, meaning the hydrophobicity of the analytes. And what we can see here for this particular solvent is that when we have very hydrophilic substances, none or very, very little is extracted until we reach a point where we have a sharp boundary that separates those that are extracted from those that are not extracted. Then we have a region where basically anything that is in this region is extracted, and then gradually the extraction efficiency decreases as substances become so hydrophobic that they are trapped inside of this liquid membrane. This means that the extract you get will be very specific to whatever is inside of this region, meaning that if we consider a blood-based sample, then all the salts and polar endogenous metabolites that will be towards the more hydrophilic region will be discriminated from going into the liquid membrane. And on the other hand, things like hydrophobic phospholipids that would be up here are discriminated from passing through into the acceptor solution. On the other hand, if we are actually interested in the substances with the log P less than 2.5, then we also have options for those. So here we have a so-called deep eutectic solvent, which just means that we mix two components together to form a solvent. Or alternatively, we can use a solvent called 2-undecanone. We may also use a mixture of these three. And we may also add an ion pair reagent, or what you might call a transfer catalyst, that is called di-2-ethyl hexyl phosphate, or DEHP. And as you can see here, this has a negative phosphate group, meaning that it can form ionic interactions with cationic analytes and assist the transfer into the liquid membrane. Now these were examples for basic substances, and of course we have other solvents that are suitable when you have anionic or acidic substances, which we will come back to. But just to show you how we can select solvents for three very different basic substances, then here we have atomoxetine, which is an antipsychotic drug that is fairly hydrophobic with a log P of 3.8. As you can see here, this has an amine group, which allows us to make this molecule positively charged at acidic conditions. Here we have methotrexate, which is an immunosuppressive drug that is fairly hydrophobic with the hydrophilic with the log P of -0.4. This has two acidic groups, but it also has a basic group. So if we make the pH of the solutions acidic, then we can have these protonated and therefore uncharged, while this will gain a positive charge. Thirdly, we have streptomycin, which is an aminoglycoside antibiotic, which is a very, very hydrophilic molecule. This is composed of three glycosides that are linked together, and also then you have modified amino groups attached. So this can have three positive charges when you have acidic conditions. And for these three substances, we would choose the solvent I showed you before, 2-nitrophenyl octyl ether for atomoxetine. For this fairly hydrophobic, but not really extreme hydrophilic methotrexate, we will choose the deep eutectic solvent with a little bit of this ionic carrier or transfer catalyst added. While for the third substance, streptomycin, we need a lot of this transfer catalyst in another solvent. After you have chosen your liquid membrane solvent, then the next logical step is to choose what conditions the sample and acceptor solution should be adjusted to. And this mainly concerns the pH. So of course we need the pH to favor analyte ionization. This means that if we take a basic substance, then we should adjust the pH to be between 2 and 3 units below the pKa of the base, and of course conversely for the if you have acidic substances. When you have biological samples that themselves have some buffer capacity, then we typically recommending diluting the sample twofold with a suitable buffer. Alternatively, you may also dilute more, but often this is not required. The sample pH often can require optimization, especially if you use some of these ion pair or transfer catalysts, while the accepted solution often will contain between 20 and 100 millimolars of pH modifier. When you look to use mass spectrometry, then of course we recommend using mass spectrometry compatible modifiers such as formic acid or acetic acid if you want acidic condition, and ammonia if you want basic conditions. Alternatively, if that is not a concern, then hydrochloric acid or sodium hydroxide or other kinds of inorganic acids also are very efficient. The next operational parameter is the extraction voltage, which is what drives our extraction. While you often think of thousands of volts when you think of electrophoresis, then in electromembrane extraction we actually manage with much lower voltage. Typically we use between 10 and 50 volts. This is also subject to optimization, but almost always the trend is as is illustrated here for our three substances. You have a very big impact of increasing voltage when you have low values, and then you get up to a certain point where there is no benefit of increasing the voltage any further. Generally, it is recommended to set the voltage at the lowest possible voltage where you still get a high efficiency. And as you can see here, even though that the substances that we look at are very different in terms of their properties, then the optimal voltage is fairly similar for all, ranging from about 25 volts to 30 volts for these three. After considering extraction voltage, we can consider the extraction time, meaning how long should you perform the extraction for. As mentioned, this is very related to the volumes that you use. So this is just three examples using the larger volumes, meaning that you have about 300 microliters of sample and accepted solution. And here you can see that you also have faster kinetics in the beginning of the extraction, and then it ultimately forms sort of a steady state where you either achieve 100% extraction recovery or you form a steady state below that where it will not go any higher. It may be that you can see that the extraction efficiency actually decreases again if you perform the extraction for too long, and this is something you see due to effect of electrolysis, which I don't have time to go into now. But as you can see here, even for something that is really hydrophilic, which often is what takes the longest, then we only extract here up to 60 minutes. If you use smaller vials holding 100 microliters, then under ideal conditions you actually can do the extraction as fast as 5 minutes. Another advantage of electromembrane extraction, which is linked to the fact that we apply a voltage, is that we can perform online process monitoring of the extraction by monitoring the extraction current. This current is a product of ions moving across the liquid membrane interface, and I often compare this current and how you can use it to how you use HPLC pump pressure when you do chromatography. This is also used for process monitoring because the pump pressure does not tell you if the separation goes good or bad, or if you have nice or poor separation, but it tells you when something is wrong with the system, either for example that you have clogged the column or that you have air in the system, which will cause a drop in pressure. Similarly, the extraction current can tell you a bit about the health state of the system. Here there are three representative current profiles where you have the extraction current, which is measured in microamperes versus the extraction time. And what you see here is that often we have a spike in the beginning, and then the current gradually decreases onto a certain level, and this decrease is then associated with fewer ions moving across the liquid membrane interface. I often get the question if you can use this profile to determine when the extraction is finished, and if you have very high concentration. In this context, very high means tens of micrograms per milliliter of analyte, then in fact yes, you can see that when the current forms a steady state, then often the extraction will be finished. But if you have a low concentration, which is typically the case in bioanalysis, then the transport of background ions supersedes the analyte transport, and therefore you cannot use this for determining when the extraction is finished. However, you can use it to evaluate if something goes wrong in the system. So if we take a look at the first two curves, then these are representative to something that looks okay. There is no right or wrong kind of profile. How it should look like is something you determine during your method development. But if we look at the third one, then you can see that it starts off by having a profile quite similar to number 2. But then after about 12 minutes, we see a spike up to a higher level of current, which is followed by a drop down to 0. And this tells you that something has gone wrong for this particular sample. It may be that you forgot to add something while preparing the sample. It may also be that you've made a little bit of a puncture in the membrane while you prepared it. There are different reasons why you can get these profiles. But the overall advantage is that you already, while you perform the extraction, can get an idea if maybe you need to be mindful of this sample when you take it for further analysis, either that you don't inject it or that you at least are aware that maybe the results can be less than desired. An advantage of the ETN12 instrument is that you can monitor this extraction current for each and every sample that you extract, meaning that you can monitor all samples simultaneously. Okay, so by now we have been through the key parameters that you have to optimize during your method development. And now I just want to emphasize that you should perform this optimization in the real sample matrix that you expect to use. So if you expect to use a plasma sample, then optimize in blank plasma. And the reason why you have to do this is because the matrix itself may impact what are the optimal combination of parameters. This is illustrated with an example here where we have optimized two parameters for streptomycin. One is the sample pH and one is the amount of ion per reagent or transfer catalyst. Here we have optimized this in 4 different matrices. One is just pure buffer, meaning there are no matrix components. Then you have urine, protein precipitated plasma, and full plasma. And the profile or the contour plot you see here show you the extraction recovery that is gained, where blue is low efficiency and all the way up to red is 100% efficiency. And what you can see here is that the optimal combination for pure buffer is about 30% DHP and about pH 4.5. But if we switch to urine, then the optimal settings of both parameters is increased. So if you were to use the same conditions that were found for pure buffer, then of course you would get a suboptimal extraction condition. Yeah. To help make the method development faster, or even allow you to skip method development entirely, then we have also made some generic methods for electromembrane extraction. These methods are made so that they will work for most analytes that fall within a certain range of charge and hydrophobicity. Just to show you how this works, then if we make a plot here where you have the analyte charge on one axis, meaning that you either have an anion or a cation, and you have the hydrophobicity of this on the other axis. If we then fill in the quadrants, then if we have a positive ion that is hydrophobic, then we have a hydrophobic base, and so on and so on. And then we have different generic methods that target each of these quadrants. Let me show you some examples. So here you see one generic system that is called B1, B for bases. This is the same system I showed you before, which is using 2-nitrophenyl octyl ether as the liquid membrane. In this generic system, you can see that you can extract most substances that fall within about log P 2.5 to about 6 with a high efficiency. In this example, we have extracted all of these substances from human plasma. On the other hand, if you are interested in some of the substances that fall within the more hydrophilic region, then we also have a generic system for this. This is called B3, and as you can see here, this has extended the window further down towards the negative log P ranges, while we actually still are able to extract many of the more hydrophobic substances. As you can see then, when we get down in log P, things do become more challenging. Here we cannot extract absolutely all substances that are within the range, but this is something that we continuously develop systems to improve the performance for. As you can maybe guess from this, there is also a system B2 that sort of gives you selectivity in between system 1 and 3. Here you see an overview of all the generic methods we currently have developed. I won't go into too much detail on these, but as you can see, for the basic substances, we have 3 generic methods. And in combining the ranges, then you can go from log P -2 to log P 6 with these methods. Similarly, we have for acidic substances systems that can go from 0.5 to log P 6. And we continuously work to improve these ranges and add more methods to target also more hydrophilic acidic substances. I will mention that you can extract substances that are outside of these ranges with electromembrane extraction, just that the current generic methods have the ranges that you see here. Of course, then all of this can be a lot of information to take in and evaluate when you have to decide what kind of generic method you want to use or should use. And because of this, we have made a web page called howtoeme.com. And in this web page, you can search a compound by its name or similar identifier such as the CAS number. In this case, I have here searched methotrexate, and this homepage will then tell you, first of all, if EME is suitable for using with methotrexate, and secondly, it will provide you with a recommended generic system to start off by. These recommendations may also be the starting point for an optimization for this specific compound or compound you're interested in. To summarize the method development practices of electromembrane extraction, then the first step you do is that you consider the ionization and hydrophobicity of the analyte or analyte you have. So generally you consider, is it an basic or acidic substance, and you consider, is it hydrophobic or hydrophilic, and then you select conditions based on that. Afterwards, you adjust the pH of the solutions. How acidic or how basic it should be can depend on the pKa or acidity or basicity of the analytes, and it may also depend on other factors such as is illustrated down here, in which case it's recommended to use a design of experiments method. Then you select the appropriate solvents. The generic methods are a good starting point. Otherwise, there is some documentation available that describe other relevant solvents to test. One additional thing that you may consider is if you need adding one of these transfer catalysts if you have polar or hydrophilic substances. Finally, you optimize the extraction voltage and time, and you record the extraction current to allow you to do process monitoring. And then just again to re-emphasize that you can try to search the compound or compound you're interested in this How to EME webpage to try to predict what kind of generic methods is most suitable for your application. Okay, so now we have been through the basic principle and method development practices of EME, and now I would like to go into a bit more details on why you should choose EME for your sample preparation. The first point I would like to emphasize is that EME is a green and efficient sample preparation technique. As mentioned previously, there is an increasing focus on the greenness or environmentally friendliness of the analytical laboratory. And in this context, EME is very advantageous because that we first of all use very little solvents and other reagents, and also that the solvents we do use are non-toxic and non-volatile. EME also offers efficient sample preparation in the sense that we do not need very many steps to perform our work, meaning that there is not much need for human intervention or robotic handling. Just to show you an example of this, then here I have taken some information from a paper that compared the extraction of amphetamine enantiomers from urine by either a liquid-liquid extraction procedure or electromembrane extraction. And as you can see here, by using the same sample volume, EME uses first of all fewer chemicals and solvents, and also we use almost a 100-fold less solvent than is the case with LLE, and we do this by only doing 2 steps if you even count the pipetting as 1 step, whereas LLE requires more steps than that. The second major advantage is that EME extracts are directly compatible with LC-MS, as we also have discussed previously. This is because the extracts, first of all, are aqueous, meaning that you don't need to do evaporation and reconstitution, which can be both time-consuming and also resource-consuming by maybe the amount of gas you need. And additionally, the extracts are very clean from matrix components, which means that we get no or very little matrix effects in the mass spectrometer, and you can decrease the amount of downtime you have to do to clean your equipment. EME also provides very good method validation data and low variability. just to show you one example, then we have here data from a paper that was published on a number of psychoactive drugs in serum. And this is then with atomoxetine as one example. As you can see here, you get a at least 100-fold dynamic range with a nice linear calibration, high recovery, and very low precision and good accuracy with limited or no matrix effects. The third advantage I will mention is that the extracts in EME are free of phospholipids, which can be very troublesome when you do LC-MS by causing ion suppression and also fouling the column over time. In this plot, you see a chromatographic trace of phospholipids that were fragmented before they went into the MS, so that we monitor all the phospholipids at the same time. And what you see here in the upper trace is a simple protein precipitation, meaning that all the phospholipids are still present. And then in the lower window here, we have zoomed in on the y-axis. Then you see still the purely protein precipitated going out of the scale. And then the red trace shows you the electromembrane extraction results, that you have no phospholipid, not even a single trace. And if we compare that to another method you might consider, which is protein precipitation and phospholipid removal by some of these hybrid type of plates, then you can see that they actually have a little more phospholipids than is the case for electromembrane extraction, while electromembrane extraction is considerably cheaper. The fourth and final advantage I will mention here in detail is that EME is very efficient at discriminating proteins and polar endogenous metabolites. And this means that you reduce the amount of interferences that you inject onto your column. So this can extend the column lifetime, and it also can allow you to run very, very fast LC-MS separations without suffering any ion suppression. Just to show you how efficient this cleanup is, then in the figure you see here, we have a chromatogram using hydrophilic interaction liquid chromatography with a UV detector. And this is from a urine sample that either was just injected directly, as you see in the black trace, whereas the green trace shows you the extract after it has undergone electromembrane extraction using generic system B1. And just to remind you, generic system B1 will target basic substances that fall within the range of about log P 2.5 to 6. So here we see that we have eliminated almost all of the interferences that have UV absorbance in the urine. We only have a few very, very minor peaks present, whereas we would get a nice and high efficiency of the extraction with B1. Finally, I will go into some example applications of what you can use electromembrane extraction for. Previously, we have discussed some examples of compounds, but here I want to show you some applications. Previously, we have seen this slide where you can see the sort of broader classes of applications that electromembrane extraction is applicable for. But just to go into a few more details, then here we have a list of applications that it has been employed for so far. Examples include extraction of pharmaceutical drugs from human plasma or anti-cancer agents from rabbit tissue. Peptide extraction from plasma is also an option, and then you can see you have different kinds of drugs or different kinds of biological matrices. But I would like now to go into these three examples in some more details. In the first example, EME was used in the determination of 12 psychoactive substances in clinical samples. This was a study performed at the St. Olav's Hospital, University Hospital in Trondheim, Norway, where EME was compared to the routine LLE method that was employed in the clinic there. The way this procedure, EME procedure, was performed was that the sample, which was 100 microliters of human serum, was first added 25 microliters of internal standard, followed by 175 microliters of dilute formic acid. Then EME was performed using 10 microliters of this solvent called 2-nitrophenyl octyl ether, and the acceptor solution was also the same dilute formic acid. Extraction was performed at 50 volts for 15 minutes, and after extraction the acceptor solution was analyzed by a fairly fast UHPLC MS-MS method. The conclusions from this comparison to the routine LLE method was that, first of all, that the EME method was compliant to FDA validation guideline requirements. Second of all, the EME performance matched that of the routine LLE method, as can be seen from this blunt Altman diagram that shows you that the results were quite similar. One additional conclusion was that EME could simplify the sample preparation process because it eliminated the need for protein precipitation, phospholipid removal, filtration, evaporation, and reconstitution that otherwise was performed as part of this routine LLE method. The second example I will provide is the extraction of pharmaceuticals from tissue samples. And in this example, we will first look at the normal or routine method, which is that you take the tissue. In this case, it is myocardium tissue from rabbits used in pharmacokinetic studies. The tissue is then freezed before it's pulverized. Then there is a buffer added to do homogenization. And then the homogenate is extracted using organic solvents in an LLE procedure, after which you can collect the solvent and do evaporation, reconstitution, and filtration before you go on to do the LC-MS step. But in this case, when the researchers tried electromembrane extraction, they found that they actually didn't have to do the homogenization step to get good results. Instead, they simply took the pulverized tissue and added the buffer and used this as the sample and extracted the cationic analytes into the acceptor solution. And of course, this is then so pure and without any particulate matter that this could be injected directly into the LC-MS instrument, thereby saving, of course, time. Specifically, the substances that were used were anti-cancer agent danurubicin and its metabolite danurubicinol, which, as you can see here, are both bases by having an amine group in this position. So they are suitable for electromembrane extraction. The procedure was that 25 milligrams of pulverized tissue containing these substances were added 200 microliters of dilute formic acid and a little methanol. Then EME was performed using a solvent called DEHPI, which I have not mentioned previously, but this is described in some documentation that is available. And the acceptor solution was 0.5 molar acetic acid, and the extraction was performed at 15 volts for 25 minutes. And afterwards, the acceptor solution was analyzed by UHPLC MS-MS. The conclusion from this in terms of analytical performance was that the EME method gave comparable performance to the reference LLE method. But it, of course, required fewer steps than LLE, including that you could skip the homogenization. Actually, EME did not need it. You got the same results if you used homogenized tissue with EME as if you just used a simply pulverized tissue. And also compared with the LLE procedure, you had improved extraction recovery and sensitivity and sample cleanup with EME. And this includes the elimination of phospholipids, unlike what you see with LLE, of course. Again, just to reiterate that if you think that you have a substance that EME might be relevant for, you can actually check this by going to the howtoeme.com homepage and search for this substance. Here you also can get more information about the principle itself and the first ever commercial equipment for EME. The final application example I will give is the electromembrane extraction of peptides from human plasma. Initially, I will say that peptide extraction is less developed than it is the case for many small molecular drug substances, but this is something that is under extensive development now. Here you see a number of peptides that we have extracted from human plasma. And these peptides were between 3 and 13 amino acids in length and had a gravy score which is related to the hydrophobicity of the peptides that also extended both towards the hydrophilic peptides and the hydrophobic peptides. And in terms of isoelectric point, these were also quite broad. So these peptides are representative to quite many different peptides with different properties. As you can see, we cannot get currently all the way up to 100% extraction recovery, but we can get sort of moderate extraction efficiency for most of the peptides. One advantage, however, is that we can analyze the peptides afterwards using a quite fast UHPLC gradient without suffering too much matrix effects. In this case, 100% means that there is no matrix effects. All right, we have reached the end of this presentation. I hope I have convinced you that electromembrane extraction will offer you green, efficient and highly selective sample preparation for, among other kinds of samples, biological samples. This you can now perform in the world's first commercially available EME instrument ETN12 provided by Extraction Technologies Norway, and this is available for ordering now. This you can use for analyte classes such as drugs and their metabolites, and also we are working on developing it for peptides and endogenous metabolites, metals and heavy metals, and a number of preparative applications. So I will say thank you for your attention if you have made it this far, and have a nice day.